Hello and welcome back to another episode of Research Behind the Image, where I, 𝗧𝗂𝗃𝗀𝗃, your Antibody Society representative, will be interviewing each of our entries for our 2025 calendar. So today I am joined by our August edition, Shaoli from the NIH. Welcome, Shaoli. Thank you so much for joining us and your beautiful image contribution to our calendar. Would you like to introduce yourselves and tell us a little bit of your work that you're up to?
First, I would like to thank you for inviting me to this presentation. So I'm Shaoli Lin, a postdoctoral fellow from the National Cancer Institute, National Institute of Health. My research direction focuses on the biology study of the cancer target, 𝗂𝗃𝗀𝗃𝗀𝗃, and the development of cell therapies against the different cancer targets.
Oh, brilliant. It sounds like very interesting work. And so is the image behind us from your postdoctoral research?
Yes. I generated this image when I was studying the biological function of 𝗂𝗃𝗀𝗃𝗀𝗃. Yeah, I can briefly introduce it. So,
Brilliant.
GPC3, so 𝗂𝗃𝗀𝗃𝗀𝗃, which is shown as green here, is also named as GPC3. It is ubiquitously expressed in liver cancer cells, and it plays a positive role in wound signaling for liver cancer progression. And 𝗵-Catenin, which is shown as red here, is one of the downstream factors for the 𝗵-Catenin for the wound signaling, and it is a key promoter for the transcription of the cancer survival genes. So in healthy cells, 𝗵-Catenin is sequestered to the cell membrane or cytoplasm, remaining quiescent and inactive. However, in cancer cells, it can be released from the cell membrane or cytoplasm, become active, and then translocate into the nucleus for cancer survival gene expression. So in our case, we want to know whether GPC3 affects the activation of 𝗵-Catenin. So I co-studied GPC3 and 𝗵-Catenin in immunofluorescence. Basically, I fixed the liver cancer cell line hep3b, and then stunned the GPC3 with our 𝗃𝗵𝗃𝗀𝗃 antibody targeting GPC3. And then permeabilize the cells and stun the intracellular 𝗵-Catenin protein with the commercialized antibody, and then nucleus with DAPI. And after that, I took the image with the Nikon Sora confocal microscope in our core facility. And then this image showed that in the presence of GPC3, in addition to the cell membrane and the cytoplasm localization, 𝗵-Catenin, they are also localized to the nucleus, compared to the GPC3 knockout cells, where the nucleus is totally absent of 𝗵-Catenin. We conclude that GPC3 is required for the activation of 𝗵-Catenin and the subsequent cancer survival gene transcription for the tumor growth. So this is what the image talking about. Yeah.
So this only happens in the tumor site. It doesn't happen normally, this translocation into the nucleus.
Right. In cancer cells, this activity is extremely high. Yeah.
Okay. No, it's beautiful image. You can definitely see it going into the nucleus in these. And do you know what actually activates this for it to occur?
Yes. So in normal cells, 𝗵-Catenin is sequestered by a protein complex named destruction complex. And after this, it could be phosphorylated and then degraded in the cytoplasm, right? But after the initiation of wound signaling by GPC3, the destruction complex could be interrupted, and then 𝗵-Catenin could be released from this complex and exert its normal function for the cancer survival gene transcription. Yeah.
Oh, no, that's really interesting. And so I'm guessing your lab has delved deeper in this, and you're going to show us some more of your research shortly?
Yeah. So the topic I'm going to introduce is the validation of glypican-3 function in Wnt3A signaling in hepatoma carcinoma. So here. Yeah, so why we focus on glypican-3? Because GPC3 is expressed in over 70% of hepatoma carcinoma, which is a major type of liver cancer. There's no detectable levels of GPC protein either in hepatocytes from benign hepatic lesions or healthy people or patients with hepatitis. So we hypothesized that the GPC3, which is a membrane-bound unconfidel antigen, is a suitable target for antibody and cell-based cancer therapies.
Yeah. Does this molecule have any impact on the survival rate in people? Is it linked to better or worse prognosis at all?
Yeah, actually, GPC3 is associated with a poor prognosis of liver cancer patients. Yeah. So this is the structure of GPC3, the cartoon that I made. So it is composed of a core protein in the middle and two hepatocellular chain on the side. So to study the biology and function of GPC3, our lab developed 3 antibodies targeting GPC3. HN3 targets the N-terminal lobe of GPC3, HS20 targets the hepatocellular chain of GPC3, and the YP7 targets the C-terminal flexible region of the protein. Further, we use YP7 to detect the expression of GPC3 in liver cancer patients. And we see that GPC3 is only stained in the hepatoma carcinoma samples in immunohistochemistry, but not normal livers. And this indicates the GPC3 is only expressed in liver cancer and specifically, and it is a specific antigen. And also YP7 is specifically targeting GPC3. So later, based on these properties, we developed antibody-based antibody drug conjugates, T-cell engager, and CAR T therapies based on the YP7 antibody. So to further identify whether GPC3 is liver tumor specific, we also stained the other organs with YP7 antibody. And we see there is no staining or minimal staining in the other organs for GPC3, indicating that the GPC3 is ubiquitously expressed in liver tumors, which makes it a promising target for us. And to identify the biology of GPC3 in liver tumors, we first expressed the GPC3 fused with GFP and observe the protein by confocal microscope. And we see that on the cell bottom membrane, GPC3 is distributed all over the membrane. And in local area, GPC3 concentrates at short bundles ranging from 1 to 5 microliters. And this is the beta-tubulin, the cytoskeleton protein. And this is the overlay of the 2 channels. And here, when you move the microscope objective upwards, we see that GPC3 not only distributed on the side edge of the cell membrane, but also localized to the intracellular part, which is later shown to be localized with Golgi apparatus, indicating that the Golgi is the vesicle that is responsible for the transportation of GPC3 to the membrane. And in addition to the GPC3 distribution, we also want to know the dynamics of GPC3 on the cell membrane. So we performed the 𝗶𝗺𝗏 assay for GPC3. Basically, we photobleached a small area of M-cherry GPC3 on the cell membrane. And then over 150 seconds, we started to record the recovery of the GPC3 fluorescence during this period. Then we can see from here, after photobleaching, GPC3 fluorescence recovers very fast in a short period within 20 seconds, indicating that the GPC3 has a very high dynamics on the cell membrane. So to further investigate the single molecule dynamics of GPC3, I performed single molecule tracking of GPC3 molecules on the cell surface. And yeah,
what microscope did you use to do this tracking? Is it also the confocal or on higher resolution?
Not confocal, it's a single molecule tracking microscope, the HiLo microscope. This is the video for the GPC3 single molecules. And we can see that for some molecules, they are moving in a very confined motion, not traveling far away from their original location. While for some molecules, they just appear and disappear very fast, indicating that they are moving in a more freely diffused way, and they may have a higher moving speed. Yeah, and then we further plotted the trajectory of those single molecules. We see that for some molecules like this one, it is moving in a confined compartment with an invisible boundary. Not moving far away from the original location. And for other molecules, they are moving in a very mixed pattern, where at the beginning it moves in a confined motion, and then it jumped to the other compartment by a free diffused pattern. And after moving for a while, it keeps jumping to the other compartment, moving in a confined motion. Yeah, so this is the understanding for the GPC3 single molecule diffusion on the cell membrane. So after knowing the biological nature,
Did you look at the Golgi? Do they behave differently in different compartments, or is similar between cell membrane and Golgi?
Yeah, we also checked the Golgi with confocal microscope first, and we see that GPC3 are moving along with Golgi. So
yes,
that means GPC3 has the same dynamics of the
Golgi
apparatus. Yeah, apparatus. Yeah, not the same as the cell membrane. Yeah. And after knowing the biological nature of the GPC3 molecules, we are also interested in function of GPC3 in liver tumor cells. So our lab member found that GPC3 is able to recruit the free Wnt molecules outside of the cells and form complex with the cell receptor 𝗶𝗺𝗄𝗧 protein. And then the formation of the complex leads to the release of 𝗵𝗵-Catenin into nucleus to initiate the Wnt targeted genes for the HCC tumor growth. And here we further modeled the complex for GPC3, Wnt3A, and the 𝗶𝗵𝗄𝗧 receptor. We found that the GPC3 interact with Wnt3A through the amino acid around phenylalanine 41. And Wnt3A interact with 𝗶𝗵𝗄𝗧 protein through the PAM motif on its thumb. And here is the rotated view for this complex. And since GPC3 is composed of a core protein and two hepatocellular chain on the side, we want to know whether the hepatocellular chain is playing a role in the Wnt signaling. So we further did mutation of the hepatocellular chain on GPC3. We found here that when we mutate only one of the chains, there is no big interference of the Wnt signaling by the mutation. However, when we mutate both of the chains, we see a significant decrease of the Wnt signaling from here, mediated by GPC3.
So there's two possibly making redundancy for each other, but we just require something there, even if it's just one.
Yeah. So this, I think these two chains may have similar roles.
Yes.
For recruiting Wnt3A or forming the complex. Yes. And then we further identified the dynamics of GPC3 in Wnt signaling by single molecule tracking with the HiLo microscope. So without Wnt treatment, we actually identified 5 confined states for GPC3, which is represented by the black bar here. And the gray bar indicates the molecules with the treatment of Wnt3A. And also, we identified a free diffused state for GPC3 on the cell membrane. And we can see from here, after treatment of Wnt3A, most of the confined states are increased, while for the free diffused states, we see a dramatic decrease for the GPC3 molecules, which are around 80%.
Wow, big difference.
Right. So with this indicates that the Wnt3A can restrict the GPC3 to a more confined diffusion motion on the cell membrane. Yeah. possibly due to the formation of the complex. And then to identify the role of hepatocellular chain, we further use the mutated construct to perform this study. And we see here, this is the molecules without Wnt3A treatment, which is represented by the red bar. And this pink bar represents the Wnt3A treated molecules. And for most of the confound states, we still see an increase for those molecules, while for the free diffused states, we see around 40% decrease for GPC3 molecules, indicating that hepatocellular chain is also important for the confound diffusion in Wnt3A signaling. However, this effect is not as significant as the wild-type GPC3. So hepatocellular chain is also playing a role here. After knowing about the complex and the GPC3 dynamics on the cell membrane, we also investigated the downstream signaling in in the liver cancer cells. We can see that here, after Wnt3A treatment, we see an increase of the active 𝗵𝗂𝐴 in the liver cancer cells, indicating the activation of this Wnt/𝗵𝗂𝐴 signaling. However, when we inhibit GPC3 with our HS20 antibody, we see a decrease of active 𝗵𝗂𝐴 here, indicating that GPC3 should be responsible for the activation of 𝗵𝗂𝐴. Also, when we knock out GPC3 in the liver cancer cell line, we see a decrease in the Wnt signaling here. And then to further identify the effect on the activation of 𝗵𝗂𝐴, I further did a co-staining of GPC3 and 𝗵𝗂𝐴 in the immunofluorescence staining. We can see from here that in the presence of GPC3, we can not only see the membrane and cytoplasm 𝗵𝗂𝐴, we also see a decent amount of 𝗵𝗂𝐴 in the nucleus here and here, indicating that in the presence of GPC3, 𝗵𝗂𝐴 could be activated and translocated into nucleus for the cancer gene transcription. However, when we knock out GPC3 here, we see that the 𝗵𝗂𝐴 only localized to the cell membrane and cytoplasm, not in the nucleus, indicating that truly GPC3 is responsible for the activation of 𝗵𝗂𝐴 and the subsequent tumor growth. After knowing the biology and the function of GPC3 in liver cancer cells, we further engineered cell therapies targeting GPC3 on liver cancer cells. This is performed by our lab members. Basically, we engineered the HYP7 single-chain FV to the CAR construct, followed by CD8 hinge, CD8 transmembrane domain, 4-1BB molecule, and the CD3. And then we produce the CAR T cells and treat the mice with liver tumors with the CAR T cells. We can see from here, after the treatment of humanized YP7 CAR T cells, the tumor regression is very significant compared to the mouse control. And
it's
also
really fast.
It's quite potent. Yeah. And also for the orthotopic model where we transplant human tumor to the NSG mice, we see that the HYP7-based CAR T cells also have a much better regression role than the control and the antibody, the other CAR T control.
So did you had 3 antibodies to start with? Did you also progress those other ones? And what structurally made the YP7 one the most advantageous?
Yeah, we have 3 antibodies. This is the HN3 antibody targeting the N-terminal lobe of GPC3.
Yeah.
And the YP7 targets the C-terminal lobe, C-terminal region of GPC3. So one of the reasons we think HYP7 is more potent is that the C-terminal region is more close to the
cell membrane.
The cell membrane, yeah.
Okay, so it can better deliver the cytotoxicity then?
Right, in that way, the tumor cells and the CAR T cells will have a more contact in the immune synapse, and then maybe release more cytokines to destroy the tumor cells.
Yeah.
Actually, we also engineered HN3 this construct, and we found that if we shorten the hinge and the transmembrane domain of the CAR T cells, HN3 can also achieve a better potency, indicating that when you bring the CAR T cells and the tumor cells closer, their cytotoxicity could be higher.
Yeah. Oh, that's brilliant. So you've got 2 viable options at the end of the screening then. Oh, cool.
Right, yeah. And for HS20, it targets the hepatocellular chain of GPC3. Although it's very specific, but there are a lot of protein containing hepatocellular chain on the membrane. So it cannot be specifically used for therapy to target the GPC3 on the membrane. Yeah.
I see.
So
yes.
this is the 2 antibodies we primarily selected for this engineering. So based on this result, we published this finding on gastroenterology, and then further move it forward to the clinical trial at the NIH. And in this image, this is my supervisor, Dr. 𝗵𝗃𝗃𝗀𝗏, the developer of the HYP7 based CAR T cells. And this is our collaborator, Dr. Tim Grattan, the physician for the clinical trial. And this is the first patient that we recruited for the clinical trials. And currently, this clinical trial is still actively ongoing. Hopefully, we can see a very promising result based on this CAR T therapy. Overall, our conclusions here and our finding is that GPC3 is a specific liver target, and it promotes liver cancer progression by activation of Wnt signaling and beta-catenin. And YP7-based CAR T therapy is effective against the GPC3 positive liver tumor. Yeah.
Oh, brilliant. So how is your work progressing after this? What's your next stages? If this is in clinical trials, are you following that, or are you working on new targets in the lab?
Actually, in our lab, we are doing both ways. For YP7-based CAR T therapy, we are further improving it by engineering it into a TCR-based cell therapy. Also named 𝗵𝗃𝗃𝗃, we are conjugating the YP7 antibody to the TCR construct. And also we coupled it with another antibody targeting the another tumor antigen, which makes it a bispecific T-cell therapy.
It sounds like you have a very busy lab.
Right.
But no, this is hugely promising with the bispecifics exploding into the field, and given the success you've seen as monotherapy in your cells, hopefully it progresses well. Thank you all again for joining me. A special thank you to Shaoli for her wonderful research, and I look forward to seeing you again in our next episode.